concept 3 in our heredity unit is all about mutations and pedigrees. So first we have to talk about how our traits pass down. We know that mutations are any change in DNA, which just means that it's changing the order of the nucleotide bases, aka like the letters in our DNA, the A's, the T's, the C's, and the G's. And this can occur in any cell of the body. So if it occurs in your somatic cells, then it's called a somatic mutation. Remember back that somatic cells are body cells, and these are only passed down via mitosis. So if you have a mutation in your somatic cells, then this is typically what causes cancer. However, there's another option called germline mutations, and these are going to occur in germline cells that are what make gametes during meiosis. So this is where you would have a mutation that got passed down to an offspring. So if two parents are completely normal and the offspring got a genetic mutation, that would be through a germline mutation. So how are mutations caused? Well, we can make mutations via mistakes made during DNA replication that happens during mitosis or meiosis or during protein synthesis. You have mutagens, which are chemicals that can cause DNA mutations. So some examples of that is radiation or UV light or cigarette smoke. And also viruses can cause mutations to your DNA. So there's two main types of mutations that we're going to be talking about, and that's going to be a gene mutation. This happens during DNA replication. This happens during DNA replication, and it causes a change to the DNA sequence. So some examples of mutations that are gene mutations are cystic fibrosis, dwarfism, and sickle cell anemia. Then you have chromosomal mutations, which often happen during meiosis, and that's going to change the number or location of genes because it's going to impact the full chromosomes, which contain hundreds or thousands of genes. So an example of chromosomal mutations are going to be Down syndrome, Klinefelter syndrome, or Turner syndrome. So let's take a closer look at gene mutations. Within gene mutations, I can kind of subdivide those into two categories: point mutations and frameshift mutations. Point mutations are where you substitute a single nucleotide for another. So for example, sickle cell anemia is caused by the original DNA nucleotide sequence being GAG, guanine, adenine, guanine, and instead the mutated sequence is GUG, guanine, uracil, guanine. So that one difference in a nucleotide base causes the protein to be made and to fold differently, which changes its shape, and we know that shape is equal to function. So point mutations are often caused by replication errors or environmental mutagens within the DNA itself. Then you have frameshift mutations. So that's where you're going to insert or delete a nucleotide. So insert, you would be adding a nucleotide. Deletion, you would be removing a nucleotide. So for example, if the original strand was ATTACC, a deletion frameshift mutation could be ATACC, where one of the T's got deleted, one of the thymines. Another example, if the original DNA was ATTACC, then an insertion could be ACTTACC, where a cytosine got inserted before the thymines. So frameshift mutations are a bigger deal than point mutations because these will affect every codon that comes after the insertion or deletion. So for example, in the deletion, originally that had enough nucleotide bases to make two codons, ATT, one codon, and ACC, a second codon. However, after the deletion, you only have one codon there, ATA, because CC is only two nucleotides, so that's not going to code for an amino acid. take a further look at chromosome mutations. So you have duplication, which is where it's going to change the size of chromosome and result in multiple copies of a single gene. So if you look at the picture on the right, your chromosome has essentially been duplicated. So you have two copies of that large purple gene that have been attached to the same chromosome. Translocation is where you have pieces of non-homologous chromosomes that are exchanging segments during crossing over. So this is an issue since they're non-homologous. It means they don't carry the same genes. So they're not making an even trade. So for example, in crossing over, normally you would be trading similar genes. So I trade you a blue eye allele for a brown eye allele. However, since they're non-homologous trading and translocation, it's changing a blue eye allele for like a heart formation allele. And that can cause some major issues because you're going to be totally missing out on a gene that's needed. And then you have non-disjunction. So non-disjunction are where chromosomes do not separate correctly during anaphase. So what happens is you have one extra or one too few of chromosomes in a cell. So this is where you have chromosomal mutations that cause you to have 47 chromosomes or 45 chromosomes instead of the normal 46. A major example of non-disjunction is Down syndrome. So this is where you are going to get 3 copies of the 21st chromosome. And Down syndrome is also known as trisomy 21 because tri means 3. So this is where you have 3 copies of the 21st chromosome. So we can see this circled in this child's karyotype, which remember karyotype is just this picture of the chromosomes here. So in the 21st pair there is 3 instead of the normal 2. Moving on to talk about pedigrees. Pedigrees are charts that are used to trace phenotypes in a family to determine whether people carry diseases or traits. So this is a picture of a pedigree. Think of it as a family tree that is tracking a particular trait. So let's talk about how to read them. Males are represented by squares. Females are represented by circles. If you have a direct line that's connecting them, then that means that they had offspring together. The offspring are going to be in that downward bracket. So for example, in this one, the original 2 people, so this would be the dad and this would be the mom, they had 4 kids. 1, 2, 3, 4. So they had son 1, daughter 2, son 3, and daughter 4. So what this is representing is that person, daughter 4, is having kids with this man here. He's not biologically related to anybody because he's not in this bracket. But he's having kids with her, so he's included. And then these would be the grandkids of these original people up here. If it is shaded in, it means that they have that particular trait that we're tracking. And if they're not shaded in, then it means that they don't have the particular trait that we're tracking. On the side, you'll have Roman numerals, which are just showing the generations. So this pedigree is showing 3 generations of this family. We can look at pedigrees and figure out what type of inheritance that is going on within this particular family for this trait. So the first type of inheritance is autosomal recessive. This is typically the most common inheritance pattern for genetic diseases because the disease is typically more rare in the family. A lot of times you're only going to have a few members affected. Males and females are going to be equally likely to be impacted by this because because it is autosomal and it's not sex-linked. So this disease often will skip generations because a lot of times people don't realize that they're carrying the trait, and so they have kids with other carriers and passing it on. So some examples of this are sickle cell anemia, phenylketonuria, PKU, and Tay-Sachs disease. So this is an example of an autosomal recessive pedigree. It doesn't impact males or females highly skewed more one way than the other. And if we notice it is skipping a trait, it doesn't have to. That's really important. It doesn't have to skip a generation, but a lot of times it might. Moving on to autosomal dominant traits. So this is a disease that's going to be super common in the family, meaning that most of the time many of the family members are going to be impacted. So since it's autosomal, males and females are just as equally likely to inherit this disease. And generally, this disease will never skip a generation because if a parent has it, then they're very likely to pass along that dominant trait to their offspring. So some examples of autosomal dominant disorders are achondroplasia, which is a type of dwarfism, Huntington's disease, and neurofibromatosis. So this is an example of a pedigree that is autosomal dominant. So once again, we don't have a large skew towards one gender or another, and it's very common and very prevalent within this family. So that gives me two good clues that it's autosomal dominant. And the last type that we can track in a pedigree is sex-linked recessive. So this means since it's recessive, the disease is going to be rare in the family, just like autosomal recessive was. But because it's sex-linked, males are going to be more affected than females. If you remember back to sex-linked traits, because males only have one X chromosome, they have a 50-50 shot of having the dominant allele and being okay, or having the recessive allele and having the trait or the disease. Whereas since females have two X's, they have three options. They can be homozygous dominant, heterozygous, or homozygous recessive and have the disease. So they only have a 1/3 shot of having the disease, whereas males have a 50-50 shot. So because of this, disease often skips generations, and affected fathers do not pass on the trait to their sons. So take a minute and think about why that is. Alright, so here is a very small Punnett square. This is a man who has the disease. So that would be his genotype because he's XY, those are his sex chromosomes, and then that lowercase letter represents the recessive allele. And then you have mom, who is at least one capital, one dominant allele because she's not shaded in. But if I'm the son, I am chromosome XY. Where did I have to get my Y from? It had to have came from dad. So that means if you're a boy, your X had to have came from your mother. So because of that, fathers do not pass on a sex-linked disease to their son because they are giving their Y chromosome to their son and not their X. So the mother would be giving her X chromosome to her son. Some examples of sex-linked traits are hemophilia, which is a blood clotting disorder, Duchenne muscular dystrophy, and color blindness. So that's why hemophilia, Duchenne muscular dystrophy, and color blindness are more prevalent in males than females. So here's an example of a sex-linked recessive trait in a pedigree. Notice that it is skewed more towards males. That's a really big giveaway that it's most likely sex-linked. So when you are given a blank pedigree chart and you're asked to determine what type of inheritance it is, these are are ways to go about answering that. So at first I need to look at distribution of males to females that are impacted. If there is a skew towards males, then I would think it's probably a sex-linked trait. And I might work through that as though it is a sex-linked trait. If it's not skewed for males, if it's slightly more females or about equal, then I know it's probably autosomal. And so what I need to do is I need to look for two parents that are the same. Either they're both shaded or they're both not shaded, and they have a child who is different. So you have two options here. You have either two parents that are shaded with a child who is not, or two parents who are not shaded with a child who is. So it doesn't matter if they have other kids that are the same as them. That's not the point. They have to have one kid that's different than them. So the child that's different is homozygous recessive. And then the parents you label as heterozygous, because that's the only way where you can have two parents that are the same with a kid who is different. And then I look and see what is shaded. So if the child was shaded, then that is autosomal recessive because they have two little letters, two recessive alleles. If the child was not shaded, then that means that it's autosomal dominant because who was shaded? The parents who have the dominant allele. So let's practice doing this. Here is a pedigree. I don't know what type of inheritance it is. So I'm looking at this and I'm noticing that I have 4 males who have this and 3 females who have this. So I'm not quite sure if this is sex-linked or autosomal yet. Since it is slightly skewed towards males, I can see if it is sex-linked and work through this and see if that works out. So, if I have a male, first of all of your squares you can put in as XY. Because those are their sex chromosomes. And then if they are shaded, it means they have the trait. So I would put a recessive allele on the X. If they are not shaded, it means they have the dominant trait. So I would put a dominant allele on their X chromosome. And that takes care of the males. So far, I don't have any issues. With the females, they are all going to be XX. And the only way that a female can be shaded is if she has 2 recessive alleles. So, if she is not shaded, I know she has to have 1 dominant allele, and then I need to figure out if I can determine what her second one would be. So with the females that are not shaded in, I know they have to have 1 dominant allele, so 1X has to have the dominant trait, and then I need to look at their offspring or look at their parents to figure out if I can determine the other one. So for example, with individual 6, she's not shaded in, however, mom only has recessive alleles to give her, and I know if I'm a girl, one of my X's came from mom. So I know I had to get this one from my dad, but which one did I get from my mom? Well, had to be one of those 2, and they're both the same. So we know that this individual is a carrier. Same thing for her sister is number 7. However, whenever I look at individual 13 on her second X chromosome, I don't know if this one came from mom or came from dad. So because of that, I'm not sure if this is a dominant or a recessive allele. So in that case, I'm just going to put a question mark on this second one because I'm not for sure. Moving on to our next practice, I'm looking at this and I'm noticing that 4 males are impacted and 5 females are impacted. So most likely this is going to be autosomal. So because of that, I'm going to look for 2 parents that are the same with a kid that's different. And I can see that here. So person 8 and person 9 are the same and one of their kids are different. So I'm going to label them as being heterozygous. And then their child as being homozygous recessive. What is shaded in? The recessive trait is shaded in. So this is autosomal recessive. And I can go about labeling everybody else. So everybody else who is shaded in is also recessive, homozygous recessive, because that's the only way to have a recessive trait. And then everybody who is not shaded in, I know has a dominant allele, at least one. And so what I would need to do is I would need to go about figuring out, can I be sure that they're heterozygous, or do I need to put a question mark because I'm not sure? So how to do that, look at their parents, look at their offspring. If either a parent or an offspring is recessive, then you know they have to be heterozygous. So for example, person 12 here, they have a parent, person 4, who is homozygous recessive. That means that they had to get a recessive A from their mom. So they did. With person 3, he gave his son, person 10, a recessive allele, so I know that he has to be heterozygous. However, with person 15 and 16, I can't be for sure because they don't have any kids listed on here yet, and both of their parents are not homozygous recessive, so I can't be sure. So go ahead and pause this and try to fill out person 2, 7, 6, and 14. Okay, person 6 and 7 would have to be heterozygous because their father is homozygous recessive. Person 2 is a question mark because none of their kids are homozygous recessive, so I can't be for sure. And person 14 is heterozygous because their mother is homozygous recessive.